Chapter 7, the nervous system. We're going to look at everything involving this nervous system. There's a little bit of review just to put some words back in your memory banks and whatnot. First chapter here looks at the building blocks, nerves, neuroglial cells. And it looks at how nerves conduct that electricity, conduct that action potential. We will look at a couple, 2 or 3 primary neurotransmitters. We'll make reference to a few others and not dive in to all of the mechanics associated with them. Predominantly, we're going to be looking at acetylcholine, epinephrine, norepinephrine. Those are the ones that we're going to be talking about the most, because those are the ones that we're going to be talking about for muscles, for obviously how the heart is going to be increasing and decreasing activity, because epinephrine and norepinephrine obviously are involved with the autonomic nervous system. So we'll be hitting on those a little bit later. Some of the other ones that we'll briefly mention, we'll talk about those in terms of pain, learning, and a few other things kind of sprinkled out, interspersed throughout the semester here. So this is our building block chapter, if you will, chapter 7. So let's look at these neurons, review just a little bit nerves, their structure, how we can classify them, and obviously the supporting cells. Of the 2, only one can conduct electricity, right? And those are nerves. Obviously, we have the brain and the spinal cord, and we'll, I believe in the next chapter, because this is a little different how they have it set up in phys class compared to how they have it set up in anatomy classes. The nervous system is how everything works and functions. So we have to talk about the nervous system first. So we'll look at the overall here, and the next chapter we'll look at the brain and the spinal cord, and we'll start looking at nerves and whatnot, and connect all these little bitty pieces together and how the whole totality of this system works. CNS, brain and spinal cord, everything extending off into the periphery is the peripheral nervous system. Cranial nerves, spinal nerves. These 2 systems have neurons, tissue that's going to conduct impulses. These are the ones that are conducting electricity. Glial cells or neuroglial cells, they don't conduct electricity. They support, protect, ensure an impulse can go, an action potential, impulse. There's a number of different ways that it can be termed, and you'll kind of catch on, I hope, as I intersperse these words commonly throughout this lecture here. Glial cells can divide, so these are going to be the cells that are actively dividing. Neurons, for all intents and purposes, they predominantly do not. There are a few neurons that can divide in that kind of hippocampus, parahippocampal region there. Within the diencephalon and the midbrain region, there are some cells that can divide. These are going to be cells involved with memory. And the window of understanding some of that is they're just starting to open some of those avenues. So there's still a lot to kind of learn here. And even some of the concepts of physiology, especially when we get to special senses, we know what's going on. We know the bits and pieces of the machinery that are doing it. But some of the exact specific mechanisms are still nebulous, grayish. We don't truly understand why, but we have a working theory and kind of a working understanding of what these cells are doing, what these cells are doing, and how they come together to do stuff. So when it comes to the nervous system in the brain, especially special senses in the brain and in the head region there, there's still a lot that we're still figuring out. So, all of this is pretty cool stuff. Some terms, I may put a handful of these terms on the exam. I don't know. I feel like you should know what these are. Interneuron, we're going to talk about it. It's just a neuron that lies entirely within the central nervous system, but it connects sensory with other sensory components, okay? Connects nerves together. So we'll see it involved with, reflex pathways and whatnot. We have sensory nerves. And I think I hit on all this a little bit later. Sensory is afferent, Motor is efferent. For me, I always use the mnemonic same or say me. Sensory afferent, motor efferent, same. So that's how I always remember it. Sensory always conduct information, nervous impulses from the central, or sorry, from the periphery into the central. Motor always sends it from the motor cortex in the central nervous system to the periphery, to an effector organ. Somatic just means you control it. Somatic means you control it. Those are always skeletal muscles. Autonomic is stuff that just happens. This is smooth muscle and cardiac muscle, and those are things that are obviously involuntary. We have ganglion, just a group of nerve cell bodies. We have nuclei, a collection of nerve cell bodies within the central nervous system. And we have a group of axons within the CNS, and we call those tracts. In the periphery, we call them nerves. But when they're running through the spinal cord, we refer to those things as tracts. So some terms that you definitely have seen before, okay? And hopefully that you will just easily recall. I may ask a few of them on the exam. When it comes to what this nervous system does, it's all based on neurons, okay? If the neurons are there and the neurons are functioning properly, the nervous system is cool. If I can't conduct a nerve impulse, the functional part, nervous system doesn't function well. If the structure of the neuron is degraded, broken down, obviously I can't conduct an impulse and the function is lost. So these are truly the structural functional unit of the entire nervous system. They do a number of different things. Obviously, the big one that we're going to be talking about a heck of a lot is going to be conducting that electrochemical impulse. It can also be called the action potential. Called the nerve impulse. I mean, it goes by a number of monikers. We'll give some fancy pants names like depolarization, hyperpolarization, and repolarization. So we'll get into some other more descriptive terms of what's going on in this neuron here in a little bit. Neurons respond to chemical and physical stimuli. When we start talking about senses, obviously we have receptors that are going to nerves that are going to respond to specific types of stimuli. Pain, light touch, deep touch, there's your physical stimuli. Those that respond to, within the special senses are taste sensations, okay? Responding to various different chemicals. Pain receptors respond to some chemicals as well. Release a number of chemical regulators. This is there to ensure that you don't twitch constantly, chemical regulator. There'll be some chemical regulators associated with the concept of memory and learning. So there's a lot of things there. And obviously enable perception of a multitude of various different things and controls the outcomes of various different things. They all have a nerve cell body. Again, sometimes that's referred to as soma. Okay. It contains nuclei, contains all, a nucleus, usually just one. Okay. It contains all the menagerie of organelles that you would come to imagine with any type of cell. It also contains these Nissl bodies. Nissl bodies are there for protein synthesis. Synthesis. And they're, a unique thing. Again, we can cluster these nerve cell bodies, these somas, into nuclei within the central nervous system. And we can clump them all together in a ganglia within the peripheral nervous system. And in the periphery, it's going to help relay information to a very specific location, or it could help to relay it to a multitude of locations. And hopefully, that sparks a memory of the autonomic nervous system there. We have very different types of ganglia and certain aspects of the autonomic nervous system that help us to be either discrete or mass activate everything. Always have dendrites and always have an axon. Dendrites 2, and axon always away. Always, always. Another thing that you should probably be aware of is that axons conduct an action potential. But dendrites do not. They conduct this thing called a graded potential. What's the difference? One travels a very long distance. And one is a very short distance. NCE. And a graded potential only travels a short distance. Okay. Short distance. Okay. Dendrites are shorter. Okay. Dendrites are shorter, so they don't have to travel as far, which is good. And we'll see when we need to generate an action potential, we need that little short distance thing. We need that graded impulse to occur. All right. If I don't have a graded impulse traveling down that dendrite and over the nerve cell body, okay, getting to that axon, an AP doesn't occur, an action potential doesn't occur. All right, so I need both of these things. I need the short distance traveler, I need the long distance traveler. And here we have a multipolar axon right here, okay, with the typical neuron that you think when you hear nerve. Okay, we have our nerve cell body here, the nucleus. We have all the little dendrites, and there can be many of them. Okay? And then we have the axon leading away. We have this region right here. It's called the axon hillock. Okay. And I'll just kind of put this here. And I'll remember to talk about it here in a little bit as well. High number of sodium voltage gated channels. Okay. High number of sodium voltage gated channels. Let me put a plus there. There we go. Okay. And that is a key piece of anatomy. Okay. Key piece of anatomy. That graded potential just travels over this short little distance to that. Okay. And as long as I make it to there and it's stimulatory in nature, magic's going to happen. And I can have that electricity travel down that axon. You can kind of see here, we have a pseudo unipolar or unipolar. Okay. One coming off. I have a peripheral branch. I have a central branch, right? The one that's the peripheral branch is going to be the dendrite, and the one that's the central branch is going to be more the axon. Okay, but still the same phenomenon. Okay, tiny little dendrites. Okay, triggers enough action to occur right there. And we'll talk about all this here in a little bit. And the impulse travels. Gets to here. Magic's going to happen again, and it's going to travel this distance by magic. Okay, there's your axon hillock. Again, it has a very high number of sodium voltage gated channels. Okay, and that's going to be very key into getting an action potential to initialize. Okay, so the axon hillock is an important thing. Um, they can form a lot of different branches. They usually just don't extend one arm out. They'll usually see a lot of branches, collateral branches extending off of an axon. So you can see a number of those. And you see a lot of that in the region where we make connections for memories and whatnot. You see a lot of branches that come off the axon and stimulate other nerves. So that's an important concept when it comes to memory formation. And we'll mention it maybe a little bit here when we look at memory, but there's so much that we're just kind of understanding with it. It's pretty cool though. Axons are going to be covered in myelin. Okay. And myelin is what?
It helps conduct electricity.
Yeah.
Without it, it doesn't. It's hard to explain, but yeah, it really helps conduct electricity. And without it, the nerve, it can still do it. It's just not as effective.
Correct. And it's dramatic how fast a little bit of fat goes. Okay. So myelin is fat. It helps to insulate the axon. Excuse me. We don't have myelin wrapped around dendrites. Right? We have it generally wrapped around the axon. We have, and axons can vary in length. They can be just a few millimeters. It can be upwards of a meter long. So these axons can be pretty damn long in the grand scheme of things. And giant squids, they basically are from like right here to about, 7-10 feet in that direction. Those are weird and creepy. And I've seen you can search them up on YouTube probably and see some videos of them doing research with squid nerves because they are so huge. You don't need a microscope to see it. It just sits right there in a big old tub. It's really weird. We have little spots where we don't have myelin, and that's awesome. Those are called nodes of Ranvier. They also have another name, which I don't know why they don't have it, because physiologists really just give the middle finger to new terms. But there is another term that you may have learned it by, so I'll put it up here. Make sure I spell it right. RAL. Yep. Nodes of Ranvier are also referred to as a neurofibril node now. It's the same thing. It's just they are trying to get away in anatomy. They're trying to get away from named nomenclature. So they're trying to get away from the dudes who discovered it. And which is cool because, there's a lot of Bowman's things. There's some Bowman's glands and a lot of different structures. And I guess Bowman was a very common name for a bunch of researchers back in the day. I don't know. But there's a lot of those structures all over, not just in the kidneys. And so they're trying to get away from a lot of that name nomenclature. So nodes of Ranvier is one of those things that they're trying to get rid of. Physiologists are just very slow to change terminology. All right. So neurofibril node of Ranvier, it is the same thing. Okay. So if you're describing this, let's say I ask a question on the exam, describe to me the action potential sequence that is occurring there. And if you call it node of Ranvier or if you call it neurofibril node, I do not care. And again, close up there, you can see all that myelin there. You can see the myelinating region, the node of Ranvier. It's pretty interesting how they have this, because in the anatomy book, I don't know if they really impress upon it a little bit, but you can have a Schwann cell there and it not produce all that myelin. Okay? And I believe I'll talk about this maybe in a little bit, but Schwann cells are always there. It's just some of them don't produce myelin to this degree, and we refer to them as unmyelinated. Question? Okay. Guess not. But we'll have a couple pictures to kind of show that off. Because these things are needy, okay, they're structures. We have to maintain this. We have to send supplies up and down the axon. And if this thing's a meter long, obviously that's going to take quite a bit of time. Axonal transport is this phenomena of moving organelles needed to a specific location, or proteins, okay, to maintain the integrity of the axon, to maintain the integrity of this nerve. This involves a multitude of various different proteins, and to be honest, the book goes into them. I'm not going to discuss those proteins, kinesins, dianesins. There are a multitude of different ones. Those are the two biggies. But we have two types of movement, all right? And that's what I do want you to understand, because I have to maintain these structures. And it's pretty easy to maintain a cell when it's just a little, square. I don't have to travel a whole heck of a lot of distance. With this, it does require a little bit of upkeep on this thing. So you have to spend a little bit of energy to get these proteins to extend that entire length of the axon, send the components to fix the supplies, pick up all the junk, and bring it back so I can pump it out and remove it. All right. So we have two types of movement: anterograde and retrograde. Anterograde starts in the cell body and extends out. Okay. And extends out. So it'll send from the cell body to the dendrites, or from the soma down the axon. And that uses kinesin, molecular motor proteins. Okay. And they're kind of weird and funky. They have kind of this weird kind of compass looking thing, and they just kind of walk across protein fibers, those microfilaments. So it's pretty easy for it to do. Some of them go really fast, some of them go really slow. It just depends on what the kinesin and the dian components are interacting with. If there's microfilaments and microtubules, that goes a little bit slower because there's just more things to interact with. If you get rid of one of those, it goes a hell of a lot faster. So, this is the reason why, some neurons take forever in a day to heal, and some may never heal, okay? Because there's such a great distance. There's the blood-brain barrier. There's these things called pericytes. There's a number of roadblocks that nerves have to get over to get the raw materials within them to upkeep them and maintain them. So if you damage a nerve extensively, that can really limit its ability to repair itself. Retrograde just goes backwards, from the dendrites to the soma, from the axon to the soma. Okay. So anterograde is generally sending the machinery to help fix. Retrograde is sending the waste back to the nerve cell body so we can get rid of it effectively. And there's a number of different ways that we can do that. And we'll briefly mention one. We're not going to get into that. We have a multitude of ways we can classify neurons according to their function. We can classify neurons according to their shape. Shape is probably the most common. Functional is probably the most easiest. We have sensory nerves. We have motor nerves. Okay. Those are your afferent. And motor send out the muscle command, or the effector command. Okay. Last one is association neurons. And whenever we have association neuron, does that mean it's going to be a faster nerve impulse or a slower nerve impulse?
Faster
Why faster?
because of the because it's taking it from the um something to do with the synapse. I don't know. So,
That's okay.
it's taking it from, it's like the I don't know, I forgot.
That's okay. You're trying. I love it.
It's like the transition mode. So it takes it from like the dendrites or the cell body and it moves it to it's something to do with the synapse or something. It's like oh.
All right. I'm going to help you out here, Jeff. Thank you. Love it. Okay. Whenever we have an interneuron, it's going to slow it down a little bit. Why is it going to slow it down? What is this thing right there? It's that thing right there. What are those two little gaps? Anybody remember?
Synaptic clefts.
Synaptic cleft, correct, on all fronts. Whenever we have a synapse involved, that's going to slow down a nerve impulse. Okay, it's going to slow down an action potential. Why? Because we have to get calcium to go into the synaptic knob. I have to have the synaptic vesicle fuse with the membrane, plasma
It
membrane.
has to like open and do all that stuff.
Release.
So that's why it's kind of low.
Attached to a receptor and yada, yada, yada. Okay. So it is a limiting factor. So if we have an interneuron, okay, or association neurons, that's going to slow down the nerve impulse just a little bit. How much? I mean, a fraction of a second. Is that a lot? Not really, but it is a delay. Okay. Because if we have a nerve that's myelinated, it's going to, and it's big, it's going to go roughly, that impulse is going to travel around 225 miles an hour, 224 miles an hour. Damn, that's fast. Okay. If it's not fully myelinated and it's small, then it goes about 2 miles per hour. Still, that's pretty quick. I mean, I'm 2 miles an hour from down. I mean, it's still pretty fast. But the limiting factor in all movement, action potential wise, is that synapse. Okay, so if we have an interneuron, that's great. It's integrating functions with multiple other spots and locales, but it does slow down and limit the speed to which an impulse can travel. When it comes to sensory, it is sensory or sensory neurons. Okay, we have various different types of sensory that we pick up. Vibration, different types of vibration actually. Light touch, deep touch, continuous touch, pressure, pain, heat, special senses. So we have a number of different various types of sensory neurons depending upon what type of modality that they pick up. And obviously, we'll expound about that when we start talking about senses. When it comes to motor neurons, there are some subtle differences between two basic types: your somatic motor neurons and your autonomic motor neurons. All right. Somatic, that means body. So again, these are going to be stuff that we voluntarily control. Skeletal muscles, it's about the only thing we voluntarily control. When it comes to, excuse me, goodness, when it comes to somatic motor neurons, they extend out into the periphery and they go to the effector. They go to the skeletal muscle, the deltoid. Autonomic usually is a relay of a couple different neurons. These are going to supply, innervate means supply, stuff that's involuntary, and that's anything else that's not skeletal muscle. Okay, I put some examples there. That's, pretty much everything else. So this is going to be a relay of a couple different neurons, and I have a picture there kind of showing this, I believe, on the next slide. We have a couple different types of autonomic motor neurons. Those that are involved with fight and flight, sympathetic, and those that are involved with rest and digest. These are very opposite, right, in how they affect different structures. Okay, what's sympathetic on one effector maybe not be affected sympathetically. Okay, be more stimulated, be more parasympathetic. We'll look at that when we start looking at the autonomic nervous system. Okay, so we'll dive into some of the how all that nonsense works, getting us ready to fight the bobcat in the parking lot, or getting us ready to lay down and go to bed, or eat a sandwich, whatever the case may be. But they do different. There's autonomic motor neurons. See, there's one, there's two. Somatic, I mean, it's just one neuron that extends out. Okay. All skeletal muscles are innervated by just one nerve. And that ensures really proper function. And it's cool. I mean, you can see this in utero. You can see in that, in those first few months of life, as they start to, as an infant starts to coordinate muscular activities, you have billions of nerves, right? And you whittle them away. Okay, you whittle them away. You see a couple different nerves going and supplying a muscle, and through reinforcement, one of those is reinforced, the one that goes to the muscle that's supposed to, and the other one that's going to it as a secondary kind of atrophies and goes away. So this is a pretty cool phenomenon that you can see, in certain studies. But there's your somatic motor neuron, just one nerve extending out to the effector, stuff that you control. Autonomic, it's a relay. It's a series of nerves. A preganglionic neuron and a postganglionic neuron. Structure, oh my gosh, so easy, right? Pseudo-unipolar or unipolar. This book uses pseudo-unipolar. Maybe your anatomy book use unipolar. Same damn thing. Okay. There's one process coming off the cell body. That's all that means. All right. Why do they refer to it as pseudo? Because even though one thing is coming, it has two branches. Okay. A central branch, that's the axon more times than not, and a peripheral branch that is more times than not associated with the dendrite. A lot of sensory neurons are this fashion. Okay? A lot of sensory neurons are this fashion. Bipolar are more your special senses. Okay? Bipolar, there's the cell body, and there's the dendrite, and there's the axon. There you go. There's the D, and there's the A. Okay, the dendrite and the axon. But again, two processes. 1, 2. 1 with 2 branches. Pseudo-unipolar. Multipolar, this is the most common type. This is the most common type you see all over. Again, usually many dendrites and axon. Okay, I guess my pictures aren't the best. Here's some better ones. Okay. Pseudo-unipolar, one and a peripheral branch and a central branch. Bipolar, your special senses, and you'll see this set up a lot when we talk about special senses. 1, 2, dendrite, axon, and then multipolar, the most common one that we know of here. There's your axon and a multitude of dendrites. You can have nerves where, if you remember the cranial nerves, you know that some were sensory, some were motor only, and some carried both. Nerves, generally speaking, carry both. Okay. When we talk about all the nerves extending out to the various different skeletal muscles, there's a branch that goes to the muscle itself, and there'll be a branch that comes off of that and supplies skin in that region over the muscle. So the vast majority of all peripheral nerves are going to be mixed. Okay. Cranial nerves are a little bit different. Some are only sensory, okay? Like olfactory, optic. Okay? Oculomotor is just motor. Right? And facial, that was mixed. They carry both sensory and motor. So cranial nerves are a little bit different. And that's just because of the special senses up there. Bundle of axons that we find within the central nervous system traveling up the spinal cord, they're going to be referred to as nerve tracts. And we'll look at those a little bit later and see what they carry.